Yes or no? Yes. Now, the hard part is, what property do they have to have? Do they have to be hydrophobic or do they have to be hydrophilic?
Philic.
Okay, hydrophilic means what? They like what?
They like water.
They like water. Now, the membrane of the rough ER, is it made of something that it can interact with water? No, because what's the membrane made of? Phospholipids. So, our amino acids have to be hydrophobic. They don't like water, so they can interact with lipids of the phospholipid bilayer. Okay, good. And then we talked a little bit about the process of replication of DNA. So when does replication happen? When? S phase of the cell cycle. Now you're cooking, right? So the S phase of the cell cycle, right before the cell is ready to divide. And the purpose of replication is that each chromosome is going to make a copy of itself, right? So that in general, instead of having how many chromosomes do we have? 46, right? So we have 23 pairs, and instead of having 23 pairs, we're going to have 46 pairs because we're going to have copies of all of the chromosomes, right? And we call those sister chromatids. So that's what we're going to create with the process of DNA replication. Okay, so the last thing that we discussed was what enzymes are. What class of macromolecule are most enzymes?
Proteins.
Proteins, right. And enzymes themselves, they do what? What is their purpose? They're catalysts, exactly. What's a catalyst?
Something that initiates change, or a process, initiates a process.
Right. An enzyme is a catalyst, and a catalyst is something that initiates something to happen, and it takes less energy for that process to occur. So in other words, something can begin using less energy, so it can occur more quickly. That's the whole idea. Enzymes speed up how quickly reactions can take place, whatever reaction that is. It could be a synthesis reaction, it could be hydrolysis reaction, whatever. So enzymes themselves, they have a very highly ordered three-dimensional shape or conformation, as we call it. And so this is where, as we had talked about a little bit before, in lab we talked about this too, is that we know that amino acids are strung together, and amino acids, when they're strung together, they have the primary structure, basically. They're like beads on a string. But you've seen in the videos that I've shown you that primary structure doesn't last long because of the reactive groups. We have all these different these molecules that are going to interact with each other, form different bonds. So we end up in a secondary structure very quickly. In the secondary structure, we have either the alpha helix or the beta pleated sheath. And then that folds even more into the tertiary structure, which for a lot of proteins is the final three-dimensional shape. Some, we said, also took on a quaternary structure. But the point is that the amino acid sequence has to be just right. You can't have one amino acid out of place. Why? Because if you have one amino acid out of place, there can be a very strange folding of the protein, and it won't be the right shape, in which case it's not going to function properly, because the enzymes all have something called an active site. They're little pockets where some type of substrate or reactant will fit that it's going to interact with to cause the reaction to occur. And so the active site has to be the right shape, and if you have one amino acid out of place, it's possible that that shape will be wrong. Does that make sense? Does that make sense to you?
Yeah.
All right. Okay, so enzymes act by bringing the substrates close together so that they can react. And so this diagram shows what this would look like. So this blue structure is the enzyme itself. These little pockets are the active sites. And of course, the green and the red little structures are our substrates or our reactants. So what's happening here is our substrates are binding to their respective active sites, and when they bind, that sets up the situation so that the two are very close together. They're very close together, and so they can form bonds, they can interact, and what will happen is they end up making product D and product C, which are completely different than what we started out with. Substrate A and substrate B become product C and product D. Can this enzyme act on product C and D, the product that it created? No, it cannot. Usually enzymes work in one direction. When we get into the metabolism, we're going to see that in metabolic pathways, there are a series of reaction. We produce different products with each little reaction that takes place. And there is an enzyme that works at every single reaction site. And the thing is that if those enzymes could act on the product that they produce, metabolic pathway would not proceed in just one direction. It could actually go back and forth, and that wouldn't be very efficient. So generally speaking, enzymes are going to work on just one substrate and produce one type of product. Now there is one exception to this rule that we're going to be covering in this class, and that is with a particular enzyme called carbonic anhydrase. And again, we're going to be taking a look at that reaction again that we looked at with the buffering system, if you remember that. I told you you should probably start to memorize what the names of those compounds are that are in that particular reaction, because we're going to see it 2 more times during the course of this class. So basically, that particular enzyme works in a bidirectional way, and that is not common when it comes to enzymes. Most enzymes only work on one particular substrate, and they can't act on the products that they produce. Okay? Because the products that are produced are a different shape than what the active site was. Yes?
If they don't turn out correct, if the 3 patterns are incorrect, they won't make the right enzyme. What if they don't make the right shape? What is the result of that?
So that's actually,
it's got to produce something, right?
That's a topic that's actually going to be covered at the end. If we have an enzyme that is not shaped properly, it is not functional, and therefore you cannot get to a final product. So, for example, we're going to see people who have albinism, for example, they cannot make melanin. So they have very pale skin, pink eyes, because there's no color to the iris. There's no color in the hair. So it's everybody that has albinism are completely very, very pale, pink eyes, white hair. Essentially, these people are very photosensitive. The sun will kill them. It's not good. Now the reason for that is that there is an amino acid that has to be metabolized to end up producing melanin. And in metabolic pathways, we have a reactant, a product produced. Enzyme makes that. And that product becomes the reactant for the next reaction. And then we get a product. Then that product becomes a reactant for the next one. And you have enzymes all along the way until we get to our final end product. And if there is an enzyme that's defective, that doesn't have the right shape at any point in that metabolic pathway, you cannot get to melanin. And therefore you have a metabolic disorder, they call it. So there are different types of inborn errors of metabolism, we call them. Yeah. So again, I think you get sort of an appreciation for just how intricate, considering that proteins are usually, 1,000 or more amino acids long, and the process happens very quickly, this whole process of transcription and translation, but it happens so precisely. It happens so precisely. We get just the right amino acids, the right place, the protein, because we have millions of proteins, millions of enzymes, and all of them are capable of performing what they need to. And if one of them were wrong, it could cause a problem physiologically, cause a pathology. So life is very, very delicate, that's for sure. All right, so the enzyme itself, in any reaction that it catalyzes, when the product is produced, the enzyme itself remains unchanged. It is not used in the reaction other than to help set the situation up so that the two substrates, or whatever, how many substrates there are, can produce the product. But then that enzyme will go back and work on the same substrate it just worked on. It itself is not changed by the reaction. Okay, so how do we name enzymes? You may remember that most enzymes have the ending ASE, or ACE. There are lots of examples of enzymes. We'll talk about some in this class. But generally speaking, when you see the ASE ending, you can pretty much determine that you're talking about an enzyme. If you see an OSE ending, like glucose, sucrose, fructose, maltose, OSE usually refers to a sugar. All, the suffix all, I'm trying to think, alcohol, but I'm trying to think. The OL ending usually refers to an alcohol. So we don't really talk too much about different alcohols in this class, but you'll start to see patterns, because the one thing I do think is difficult, and I really do understand this, is that the vastness of the vocabulary that you need for a class like this is sometimes very overwhelming. That alone takes time to understand, let alone understanding the concepts that you use the vocabulary with. And so I'm trying to give you some general parameters in terms of when you're looking, like we talked about lysis. If you see that ending, hydrolysis, glycolysis, we're going to be talking about, you're talking about breaking something apart, right? So try to look for those types of endings or beginnings, depending upon what we're talking about, because that will help you to decipher a little bit more what the words are. So ASE is an ending for enzymes, and in general, we'll see the word phosphatase. For example, phosphatases, there are different ones. Phosphatases in general remove phosphate groups from molecules. Kinases add phosphate groups. So if you see that. The other thing, it's kind of like, actually, it's sort of like naming epithelium. You always had to put your arrangement first and then your shape after. So like simple, squamous, right? So you had two things. In naming enzymes, you can put like phosphatase in the name of the enzyme, kinase, and that'll tell you what the enzyme does. So it can remove a phosphate, kinases can add a phosphate. The other thing that you can add is what the substrate is that particular enzyme acts upon. So for example, we're going to be talking about an enzyme called glucose 6-phosphatase. This enzyme specifically works on glucose 6-phosphate. So that's its substrate. And it's a phosphatase because it removes phosphates. Does that make sense? We're going to talk about that, and we'll talk about it more specifically in metabolism. So the name, again, can specify the substrate of the enzyme. Another example would be something like lactase, which acts on lactose. People who are lactose intolerant, they don't have lactase in their digestive tract, or maltase, which acts on maltose. So again, it can have that, and that makes it a little bit easier. So you know what it does, you know what it acts on, or what it does, or both. Dehydrogenases remove hydrogen atoms. You see the prefix D, that means to take away. Hydrogen, so hydrogen is, of course, hydrogen, and it's an enzyme, so it's ACE. So a dehydrogenase removes hydrogen atoms. An example of this is lactate dehydrogenase. So lactic acid, which is actually generated in skeletal muscle, it can have hydrogens removed from it by lactate dehydrogenase, which makes it pretty much unable to donate hydrogens. So you don't want an acid anymore, right? Does this make, yes?
I just have a question.
Yeah.
About people being lactose intolerant. Does that change over time sometimes?
Not that I've really heard of, but lactose intolerance is interesting in general, because technically human beings, like after, you know, we're infants and we can start eating solid foods, you don't really need to rely on milk anymore. So one of the things you'll find is people who have come from, you know, have heritage in countries which are able to harvest twice a year, so like warmer climates, like climates in Africa, for example, or in Asia, or trying to think where else, Spain, trying to think where else, warmer climatic places. Those people tend to have lactose intolerance issues more readily than people who, like me, come from Eastern European countries where you get cold, harsh winters, right? You can't harvest twice a year. You got one harvest season, that's it. And in that case, you have to rely on your dairy. So kind of in an evolutionary way, people who lose lactose tolerance are not going to be able to survive in those climates. So people generally have lactose tolerance for longer in their lives.
I was just wondering if it changed because
Somebody.
I'm sorry.
No, somebody
I just,
did.
My mom, as she's getting older, is now lactose intolerant. Like she's drinking milk and it hurts her stomach. She can't drink it. And she drank it the other day, regular, like it's 1%, but it's still regular milk. I said, Mom, you can't be drinking that.
Yeah.
But when she drinks the lactate, she's okay. Like I'm like, you no longer can have dairy. Like
Yeah.
again.
Well, you
I
can.
just wonder, like, well, she can't drink milk. That's for sure.
If you take that medicine, huh? You have the powder. I know what you're talking about. Yeah, I know what you're talking about. My lab assistant was lactose intolerant, and she was like her parents were Italian.
I just
So
wondered, like, what changes that?
what changes it? Well, again, generally it depends.
It's real depth. You don't have to.
No, I mean it's a good question. I mean it does depend. I mean a lot of people who are lactose intolerant, they might lose the tolerance after infancy, but some people it does. It takes longer for it to happen. But I mean the good news is at least you can take some medicine. Like Shannon used to take medicine and then she'd be able to eat like pizza or something like that. She'd be okay. But yeah, I know it's really tough because
secret gene that pops up. You know, from the last slide on the glasses.
yeah,
What's it called?
epigenetic inheritance. So you did study that. You studied.
Because I was trying to watch many videos, but I couldn't still understand, like, so does it, something that doesn't occur, like say for example you have lupus.
Yeah.
Is there a way you can alter it and switch it? If that gene wants to go back off or whatever, I was totally like.
It generally no, but sometimes like certain with epigenetic inheritance, certain, like you could have two people who are twins and they have the same chromosomal makeup. And the one person doesn't develop the particular condition, the other one does, and it could be because maybe that person is under more stress, even though they're identical twins, one gene could be muted for longer. That's what epigenetic means. Epi, like to stand on it, so it's like you stand on a gene, you're not expressing the gene. But then, like the other twin may be under a lot of stress, so they do express the gene. That is, yeah. That's a good question. Good.
Yeah, I can't drink milk in my coffee.
Mhm.
I have to use half and half or something. The milk upsets my stomach.
Yeah. Some people it does that. Yeah. And sometimes it takes a while to become lactose intolerant, but now there's so much, out there like Lactaid and soy milk, almond milk, all that. It's like, it's nice. It's very nice. No, I drink it all the time. I'm not lactose intolerant, but
It's just delicious.
it is good. I like it. I love it.
Yeah, I like almond milk.
Huh?
I like almond milk.
I do too. I like it like if you're making desserts or you're making like desserts or curries and stuff like that. Like it's good flavor. All right. Anyway, so now this diagram, or I should say table, is showing us some names of some of the I guess important enzymes. Some of these we'll be talking about. Some of them we will not. Carbonic anhydrase I mentioned a little bit earlier. We'll talk more about that. That helps to catalyze the reaction. Does anybody remember what H2CO3 is? Carbonic acid. Awesome. You guys are good. Carbonic acid to water and carbon dioxide. Now it's only showing it going in this direction, but interestingly, and this is like, was that me or was that you? Oh, that was you. Kirsten. Making me think that it was me dropping stuff. Gosh. It's trying to mess me up. Anyway, carbonic anhydrase is a very interesting enzyme because it's one of the very few enzymes which can catalyze a reaction in the opposite direction. So it can also catalyze the reaction of water and carbon dioxide going back to carbonic acid. And again, I mean, what's nice about the naming of enzymes, you've got carbonic in there for carbonic acid, and it's anhydrase. Because what it does is it actually takes away some of the hydrogen, right? So an means take away, hydrase is the hydra, I should say, is the hydrogen, and ace is the fact that it's an enzyme. Amylase is in the small intestine, and it helps to actually take starch and water and break it into maltose, which is a monosaccharide. And again, like, with lactase that we were just mentioning, lactose, if you remember, it's a disaccharide, right? It's made up of glucose and galactose. And ultimately, you can't, in the small intestine, absorb a disaccharide. You have to break it into monosaccharides. And then that can be absorbed through what I showed you guys on that diagram, through the processes of primary, secondary active transport, facilitated diffusion. Glucose molecules can come into the bloodstream. So if you don't have lactase, what the problem is that the disaccharide stays in there and the bacteria eat it, and then they start to produce gases and they can cause other issues. So that's why people have issues with pain, bloating, diarrhea, those types of things if they're lactose intolerant and they take in milk. Okay, so those are some of the enzymes that, again, we'll be talking about. Now, enzyme activity, how quickly an enzyme works. So, in other words, the rate that substrates are converted to products. Again, how quickly an enzyme can do this is affected by a couple of different variables. So we're going to skip temperature for a second and pH. First of all, we'll talk about number 3, the concentration of the enzyme that's available to catalyze the reaction. So obviously there's a directly proportional relationship to this. The more enzyme we have available to catalyze a reaction, the more quickly we're going to convert substrates to products. That makes sense. Now, temperature is tricky. We talked a little bit about the influence of temperature before, but temperature, as we increase the temperature, what do you think will happen to the rate of how quickly the enzymes work? You think it'll go up or down?
Speed up.
Speed up, right. Well, I guess it's sort of like when you think about the weather, right? Whenever the weather starts to get warm out, don't we feel like going outside and doing more things more quickly, more efficiently, right?
Depends on how hot it is.
But, exactly. As the temperature gets really, really hot, like yesterday, for example, what happens? We start to slow back down. Basically, we can't just increase the temperature into perpetuity and keep on making the substrate be converted to product faster and faster and faster and faster and faster. You can't do that. Because with proteins, especially, which enzymes are, we know that if we get too high of a temperature, what happens? What happened? Well, it slows down, but the reason is, as Kirsten said, because it denatures the proteins, right? So they unfold and the enzymes can't work anymore. So we can increase the temperature to a point and we increase how quickly the substrates are converted to products, but only to a point, after which we start to decline rapidly. And pH. Now pH is tricky because enzymes work in all sorts of environments, not just in the bloodstream, like carbonic anhydrase. Carbonic anhydrase works in the blood, so it's going to work really well at a pH of 7.35 to 7.45. But we know that all through the digestive tract we have various enzymes that work in different locations. So for example, in the mouth, does anybody remember what the enzyme was that we have in saliva? We have salivary. It breaks down sugars. It starts to break down sugars. What? What'd you say? I don't think. No. What did you say?
What does it start with?
Oh, what does it start with? Oh, I thought you said something else. I was like, I don't know. A. And it's actually on the previous diet. It was on the previous table that we found. Amylase, right? Salivary amylase. It starts to break down sugars. So the saliva is pretty basic, all right? But in the stomach, we have another enzyme that's found there. Now this one doesn't have an ASE ending, but it is in a very acidic environment. You know, the pH of the stomach is like 2, right? And so this enzyme, it starts to break down proteins, and it's called pepsin. Pepsin works very well at a very acidic pH, and it is specifically designed to be able to withstand that so that it does not denature. So here we can see in this diagram showing the effects of temperature and pH. Temperature, again, will increase the enzyme activity, but only to a point. After we get too hot, enzyme activity declines rapidly because of denaturation. Enzyme activity at different pHs, again, it depends on the enzyme. Pepsin in the stomach works very optimally at a pH of 2, which is good because that's the pH of the stomach. Salivary amylase works more around a pH of, if we look, it's about a pH of 7 or so. And trypsin. Trypsin comes from the pancreas, and we know the pancreas secretes its secretions into the small intestine. And in pancreatic juice, we have pancreatic trypsin, pancreatic amylase, pancreatic lipase, break down sugars, proteins, fats. respectively. And we also know that pancreatic juice contains bicarbonate, which is very basic. So trypsin works really well at about a pH of 9 or so, maybe a little bit higher. So it shows stomach pepsin works well at a pH of 2. Salivary amylase works well at a pH of almost 7. Trypsin at a pH of 9.5, very basic. So again, optimal pH ranges are going to be different for different enzymes. Okay, so cofactors and coenzymes. Let's talk a little bit about this, and then let me see how many slides we have left, see where we take our break. I think what we'll do you guys need a break, you think? You need a break? No? Yes? Go home, find out. You're going home. All right, you think you can just hang in for maybe like 5 more minutes, 10 more minutes? We take like 10 minutes and then, okay, that'll be a good halfway point. All right, so talking about cofactors and coenzymes, these guys are necessary for the normal activity of enzymes. So they help enzymes out, basically. And coenzymes, we are going to be talking about with glycolysis and the metabolic pathways. They're really not explained too much here, but essentially these are going to be derived from vitamins like niacin and riboflavin. And there are two ones that we're really interested in, NAD and FAD. NAD and FAD is what we call them. And they basically are like letter carriers. They carry around 1 letter, and that's H, which is hydrogen. So that's their big job. Cofactors are what is described pretty well here. These are things like metal ions like calcium, magnesium, copper, zinc, and selenium. And they work in different ways. And this diagram shows a couple of the different ways that they can help an enzyme out to do its job. So cofactors, when they bind, they change the conformation of the active site. So a lot of enzymes, for example, the pancreatic enzymes, okay, like trypsin and lipase and all of that, they're going to be secreted in an inactive form. So trypsin, when it's created in the pancreas, is known as trypsinogen. In the stomach, pepsin is known as pepsinogen. But when it hits that acidity of the stomach or the acidity of the small intestine, that's when they become pepsin and trypsin. And part of the reason for that is that the binding of cofactors activate these enzymes. If these enzymes were always active, the pancreas, the stomach would digest themselves, and that wouldn't be so good, right? We don't want the enzymes always working. We want to regulate when enzymes work and when they don't. So cofactors help with that. So cofactors, they can bind to enzymes, like in this case, they're showing a cofactor binding to another site that isn't the active site. And what that does is it causes this active site to change its shape. So now the substrates can actually bind. Likewise, in this case, it's binding to an area in the middle of the active sites so that the two substrates can more easily bond and more quickly bond together. So cofactors are going to help the enzyme to work more efficiently and more effectively. Make sense?
So they come from, they go into a non-active site and make it an active site?
They can activate the active site. It's a lot of action there. Activate the active site. That's pretty. Is that a case or is that a folder? Say, I've been like looking at it the whole moment. I'm like, oh, it's like mesmerizing. Okay, so so lastly, and then we'll take our break. So A lot of enzymes, as I mentioned earlier, are produced in an inactive form. So the pancreatic enzymes, as I said, they're not activated until they reach the small intestine, because if they were always active, they would basically digest the pancreas. So it protects the pancreas against autodigestion or self-digestion. A lot of them are activated by phosphorylation and inactivated by dephosphorylation. So again, we can actually regulate the enzyme activity, which is very important. Others can be activated by second messengers, like, for example, cyclic AMP, CAMP, C-AMP, or let me say G proteins, those types of things. And enzymes are regulated by the turnover of the enzymes. So basically, breakdown and resynthesis of enzymes. We regulate how much of an enzyme that we produce, so that we don't have too much for a particular thing, we don't have too little for a particular process. We want to have just the right amount. And once these enzymes are done with their job, a lot of them are going to be broken down. So this is where our transcription and translation comes into play. We're going to increase the amount of enzymes that we produce. We're going to increase the number of enzymes that we're going to produce, as long as we have an active process and an active need going on. But then after that process is completed, then those proteases, like we talked about, they're going to break down those proteins. Remember proteases? That's an enzyme too, that specifically break down proteins. We talked about this in the last class. So essentially, those are some of the key points about enzymes. So we will, I think, pick up here with the effect of substrate concentration on enzyme activity. When we come back, let's take 10 minutes now, so it should be about 10:15, let's come back at about 10:25, and then we'll pick up with this, and then we have just a little bit more to go.